Tinder, Bumble, Grindr, and Hinge. The matchmakers of today come as nice, neat sets of algorithms. Codes developed to distill you into a searchable, matchable set of criteria. But these codes are not all the same. Each app processes your information a little differently, and depending on your age, status, and situation, there might be one app that works best for you. Well, in my work, I'm trying to find the perfect matchmaker to bring two carbon atoms together. But before we get started, we need a little bit more information. So let's start from the beginning. Hi, my name is Erin Taylor, and I'm a graduate student in the chemistry department. And in my research, I'm trying to make a designer carbon. You may know some of its famous cousins like fullerenes or graphene, both of which won a Nobel Prize after their discoveries. Now, fullerenes and graphene were both predicted to exist decades before they were actually made in the lab, and many scientists were skeptical about the reality of one day holding these theoretical structures in our hands. Now, fullerenes are little balls made out of carbon that have the potential to destroy cancer cells. And graphene is a flat sheet of carbon that is 200 times stronger than steel while being lighter than a sheet of paper. These materials were first predicted in 1991. But there's still a missing form of carbon that scientists have predicted should exist, and it looks like a sea of infinite twisting pure carbon. These materials were first predicted in 1991 and are called Schwarzschites. Scientists have predicted some amazing and unusual properties of these materials. For example, if you were to stretch most materials, like a rubber band along its length, it gets skinnier in the width. Well, Schwarzschites would do the exact opposite, where if you would stretch them along their length, they'd also get thicker along the width as well. Now, there's also a class of materials called energy absorbers, which includes Kevlar, which is used in bulletproof vests. And the idea behind these types of materials is that they can take incoming energy, like the energy from an incoming bullet, and dissipate that energy along the material itself, meaning that the bullet can't do as much harm. Well, Schwarzschites are predicted to outperform both Kevlar and many steels with its energy absorbing properties. Now, in reality, Schwarzschites are the Tinder unicorn of the material science world. They're sexy, intriguing, and elusive, and we've never encountered them in the lab. And the weird thing is they should exist. Many scientists have performed these calculations over and over again, and these materials are predicted to be even more stable than diamond. So from what we know about trying to synthesize these materials, they aren't going to form on their own. So instead we need some mold to build them on top of. Now, coincidentally, nature has created a mold in the form of a mineral called a zeolite. And these minerals have the exact structure we want our carbon to take. So we can take a zeolite and lay down a layer of carbon on top. When we remove that zeolite, we have a freestanding carbon framework that we call zeolite templated carbons. And this is arguably the closest we've ever gotten to a real world Schwarzschites. However, zeolite templated carbons don't look as perfect or behave as bizarrely as we know Schwarzschites should. We've been catfished. And this is where my research comes in. When I first stepped foot into the lab, my advisor showed me how to make these materials. And surprisingly, it was the same procedure we've been using for the past 30 years, ever since zeolite templated carbons were discovered. All this time, we've been using the same zeolite mold. And I have a hunch this is why we aren't making Schwarzschites when we make zeolite templated carbons. So inside of the zeolite, there are thousands of little matchmakers, which are called catalytic sites. And this is where the carbon atoms can bond to each other. Now, while these catalytic sites are strong enough to form the carbon-carbon bonds, they aren't strong enough to hold the carbon to the zeolite surface. So because of this, we will never make a Schwarzschites out of a zeolite templated carbon. That is, unless we can get the carbon to stay on the zeolite surface. So we need to narrow the search radius. Instead of trying to match with someone in New York City, I'm instead going to try to narrow my search so that I can match with someone near me, like in Bozeman. This would not only allow me to create the carbon-carbon bonds, but keep that carbon very close to the zeolite surface as well. And for the past 30 years, the zeolite we've been using has sodium ions as the catalytic site. But I started to wonder, what if these sodium ions are too big that they don't allow the carbon to actually form on the zeolite surface? Well, what if we could use a smaller ion? And I have a smaller ion in mind, and this is lithium. So if sodium was the size of a cantaloupe, lithium is the size of a grapefruit. And by using lithium as my matchmaker, I hope to decrease the distance between the carbon and the zeolite. And maybe this will be the key to finally finding the one, the Schwarzschild. Thank you, and I'd be happy to take any questions.
And everybody feel free to unmute yourself to ask as well.
How close are you to making it?
That's a great question. Thank you, Mallory. I don't know how close I am yet. I'm still in the middle of trying to make the carbons themselves. Making the lithium zeolites has been quite a challenge. So I'm just starting to see how the carbon's playing a role in all of this. So hopefully soon.
Is lithium difficult to work with? I don't work in a chemistry lab, so I don't really know what that looks like. But
yeah, the type of lithium I use is a lithium chloride salt. It's very similar to sodium chloride, like table salt.
Okay.
So it's quite easy for me to use. It's just, it's so reactive that it's hard to get into a zeolite.
Okay, cool.
Thank you. All right. Well, maybe I'll pass it on to the next person. And I forget, is it?
It's me.
Galip? Okay.
Yeah, I don't have any question. And if nobody has any question, I can go. Can you hear me clearly? Okay, thank you. So, hello, this is Galip, and I'm a third-year graduate student in chemistry department. And today, actually, we'll be talking about a story of two best friends, Alex and Cody. And Cody's birthday is coming, and Alex wants to make a giant Jenga for him. But obviously he will keep this giant Jenga in his backyard. And so Alex needs to put some wax on these tiny pieces, or actually giant pieces, just in order to protect them from the effect of water, like that comes with rain and snow and so on. But if we have something that we want to protect from water, from the effect of water, do we always just put some wax on them? Actually, no, we do not do the same thing all the time. But the things that we are going to do, the process that we need to do is going to be very similar to that. And in my research, we are working with a mineral called gypsum. And gypsum is composed of some particles that can be harmful if we and animals drink some water coming with these particles. And we call these tiny particles molecules, actually. And we are trying to find some molecules that do not like water, but do like gypsum. So they will attach on gypsum surface, and they will repel the water molecules away. So when water interacts with gypsum surface, they will not steal these harmful particles or these harmful molecules from gypsum surface. But we are talking about very tiny particles that we cannot see with our eyes at all. So how are we going to work with them? Well, we use some fancy machines and we use light. So let's go back to our friends, Alex and Cody. And they always go to a nice green park on Saturdays with beautiful green color everywhere. But when we say the grass is green or tree or the leaf is green, what do we actually do? So let's go a bit deep into this and let's try to analyze what are we doing here. So white sunlight hits on the leaf and it only reflects the green color. And this green color comes to our eyes and our brain analyzes this and we say that the leaf is green. So what we do here during this process is actually called a spectroscopic analysis. And very similarly in my research, I am using a light source. I am using a laser as a light source, but not a sunlight. And my sample is gypsum, but not a leaf. And I am using a detector and the PC as our eyes and then brain. So we shoot the laser light on gypsum surface, and some of the features of this light changes, and we track these changes of light. And this is how we get some information from the gypsum surface at the molecular level. But there is one more big feature of the research technique that I'm using. It is able to tell us what plane these molecules are dancing on. So let's imagine Alex and Cody go to a dancing night, to go to a bar for a dance night after a very long day in the park, let's say. But it seems like there is a different kind of dance night tonight. And like there are two types of dancers. Like one type is dancing with both of their arms up, showing up, and the other type are dancing with both of their arms parallel to the ground. So in order to dance together, obviously same type of dancers should match with each other, right? But it is not only human beings and animals who are dancing on Earth. Molecules and light also dance, believe it or not. And so in my research, again, let's go back to my research. When we shoot the light and we use some optical components to decide on which plane our light should be dancing. So when it hits on the molecules on gypsum, it will only interact with the molecules which are dancing on the same plane. And we will see a huge increase with the brightness of the light. And at the end, we say that our molecules that behave as wax on gypsum surface, oriented like this or like this. And if they stay upright like that, it means that they will push water molecules away in a more effective way. So, okay, we shoot the light on gypsum and get some information and so on. But can we only work with gypsum by using the research technique that I'm using? Actually, all of my study is just a tiny part of huge surface chemistry field. Let's say you are eating food somewhere in a restaurant, and let's say you just check the menu. So when you check the menu, as we mentioned earlier, some light beams come to your eye, and in the neurons, some molecules are being produced. And these molecules are being transferred from one neuron to other, from one neuron to the another, and one neuron to the other. And that happens like maybe millions of times. And all of these steps, all of these transfer of these molecules are happening through the surfaces of these cells. And there is some surface chemistry happening. Or let's say you're in a research team that are designing a huge rover that you will send to Mars in a couple of years, let's say. Some of the people in your research team, or maybe you need to know that the atmosphere of Mars is not the same of atmosphere that we have on Earth. So you need to put some paint, or you need to coat the surface of your rover. So these unknown molecules, they are not going to be harmful for the surface of your huge rover that you designed and maybe spent millions of dollars. So, I mean, my research is, again, as I said, can only be considered as a tiny fraction in surface chemistry. But hopefully it will give a very useful theoretical background about how we may be able to deal with the water quality, especially in the Montana region. And as we can see, like surface chemistry is literally happening everywhere, from tiny cells in our body to a huge rover that you will send, or that is already on the Mars. And it's a vast field that you can easily find some points that you can be interested in, and maybe you can resolve a huge mystery. Thank you, and I can have your questions as well.
I was
Thank
just
you.
wondering, oh, go ahead.
Oh, I was just going to say, thank you so much, Galip. My name is Kent Davis. I'm one of the faculty members that work with these graduate students to help them. They've basically spent the last year working on storytelling skills. And so these talks may seem a little bit unfamiliar to maybe listeners or people watching them, because we're asking them to go totally solo, free-falling without any sort of traditional slides or other sort of PowerPointy sort of things, and to tell you stories and to talk to you about the science that they do. So one of the parts of this thing for all of us is that we genuinely like talking about the science that we do, and any questions you might have about the work that these folks are describing will be great and exciting for us to hear. And also, if you want to type into the chat, that's also fine. If you want to deliver some questions that way, as opposed to unmuting yourself and, I don't know, letting your dog barking come into the space, that would be awesome. Also, I don't think it'll break WebEx, but anybody who wants to like unmask, that's also totally fine. But sorry, I just wanted to say that and get that onto the open, so that in case anybody had any things that were even tangential to Galip's talk, the idea of surface chemistry or anything like that, please feel free to weigh in. Part of us talking about this stuff is that we actually want to hear from you, and these folks want to answer your questions, and they want to nerd out on their science. So like anything you want to ask, please feel free.
Thank you, Kent, for that explanation. And then I did actually have a question. If the field of surface chemistry is so vast, how did you land on water quality?
Oh, that's a great question. So what I'm doing in my research is surface chemistry happening on gypsum surface. Does that answer your question? Kind of.
Yeah, and then I guess going off of that, how did you narrow that down as your study?
Perfect. Great question. So, okay. Firstly, I am trying to understand how water dissolves gypsum at the molecular level. So like, water molecules like H2O, they may approach the surface like maybe in this way, or maybe in this way, maybe in this way. Maybe they are approaching by using their oxygen or maybe hydrogens. So I am firstly trying to understand how these water molecules dissolve this gypsum at the molecular level. And in the second place, I will try to find some molecules, as I said, which likes gypsum surface so they can attach. And in this case, these molecules will likely have a polar head group, because gypsum surface has some charges and has different charges on different surfaces. So I am trying some anionic surfactants, let's say. Surfactant is a molecule that has a head group, that is a charged head group, and with a long tail. And these long carbon chains, as you may know from these oil molecules, oil just repels water. And these long carbon chains also repel water molecules. So I'm trying to find some molecules that will attach on gypsum surface by using their head groups, but at the same time, they will stay up like this. So they will repel these water molecules. And we are trying to, like in summary, we are trying to modify the gypsum surface to suppress the dissolution of itself.
So, Galip, this is Kent. I think too, I don't want to put, I think too, maybe Cassie's question, part of it that I found interesting is, how did you end up studying what you're studying right now? Like, what was the process? Or like, did it come to you in a dream?
Well, okay. So this research has funding. I mean, I didn't write this project. It was not me who wrote the funding application or anything. So, oh, but yes, I knew about this project when I got accepted to grad school and I was talking to professors. And that was interesting. And I also love working with light. And like most challenging part in my research, I can say that is actually getting this light interacting well with the sample that you are using. So I like doing that. And yeah, that's why I chose this research. But as I said, no, I didn't write about this project.
Excellent, thanks. Oh, and there's a question in the chat as well. For many scientists, is a blend of our curiosity and a series of opportunities and trying to make the world better. This is from actually Craig Ogilvy, who's the Dean of the Graduate School. Excellent. Thank you, Dean. Any other questions for Galip?
Isn't necessarily a question much of research, but I was curious of where you were originally from.
Oh, I'm from Turkey.
Oh, I'm from Turkey. Okay.
All right, excellent. Well, I think we're going to move on to our next speaker, if that's okay, Galip. Yeah.
Yeah.
Okay. And our next speaker for today, our next storyteller, is Mallory Thomas. So take it away, Mallory.
Thank you, Kent. Hi, my name is Mallory. I am a second-year PhD student in the microbiology department. And today I want to talk to you about how the biological world that we live in is actually this ruthless game. It is a never-ending competition for the resources that we need to survive and reproduce, where winning is defined by how long you can stay in the game. Sometimes the plays of this game are quick and direct, like when two animals fight over a territory that contains food and water. But others are slower and more subdued, as opponents quietly survey one another, identifying weaknesses before striking. You and I and every organism that we share this planet with are all playing the same game against one particular rival. And this game that we play is kind of like a one-sided capture the flag, where our side is trying to protect something that our opponent desperately wants. This thing is so precious to them that their life depends on it. And because of this importance, they are willing to obtain this thing by really any means necessary. And the dark side, of course, is how destructive those means can truly be. The adversary that I am referring to is the seemingly infinite population of viruses that inhabit our planet. And the resource that they are constantly trying to obtain from us lies within every single cell in our bodies. It is the ability to turn genetic material into proteins that viruses use to replicate. Now, remember, genetic material is essentially the code of life. It's the instructions or the blueprint for how every living thing on this planet operates. And just like viruses need instructions in order to operate. But unlike us, viruses don't have the machinery that they need to take these genetic blueprints and turn them into the proteins that essentially run the world. Which is why the goal of a virus's life really is to infiltrate our cells, hijack this cellular machinery, and replicate their population. My PhD research is aimed at trying to understand why some viruses are more successful in this pursuit than others. And today I want to introduce you to a group of viruses that seems to dominate this game. and how my research is working to characterize the strategies that they use in this success, so that maybe we can better understand how to protect ourselves and the resources that they are trying to take from us. So to step back a little bit, viruses are the most abundant biological entity on this planet. There's about 10 million viral particles found in every milliliter of ocean water alone. And because of this abundance, they are also the most diverse group of organisms on our planet. Pretty much every single cell on Earth has some kind of virus that is able to infect it. But because these viruses can't survive on their own, biologists are not really sure if we should consider them to even be alive. So we should really think of them as just these little biochemical packages that are designed to carry around genetic material until they find the right cell that can replicate and repackage that genetic material. And one of the first ways that we classify viruses is by the type of genetic material that they carry. So I'm studying viruses that use RNA instead of DNA molecules as their genetic material. These RNA viruses are the most genetically diverse group of viruses on the planet, and that's because the protein that they use to replicate their genome doesn't have any kind of proofreading mechanism, which means that every time they replicate, their genome undergoes some kind of genetic mutation. And these mutations can be thought of kind of like cheat codes that allow the virus to adapt new and more efficient ways to hijack their host cells. So basically their populations are constantly changing, and sometimes they even change so much that they are able to a new species of host. These viruses that jump from host species to another are called zoonotic viruses, and they can be especially dangerous because their new host may not have encountered a virus with the same abilities before, and because of that, doesn't have any established defensive strategy to protect themselves against these kinds of viruses. This would be kind of like if the middle school JV hockey team suddenly went up like the winner of the Stanley Cup. Like, they have no chance. And human populations have experienced this defenselessness against RNA viruses with zoonotic potential multiple times just in the past 20 years alone, from two virus families in particular, influenza viruses, and then of course, coronaviruses. So in 2009, there was an influenza virus, H1N1, that jumped from pigs to humans, causing the infamous swine flu. And of course, there have now been multiple different coronaviruses that jump from bats to humans through some kind of intermediate animal reservoir, all the way back in 2002, again in 2013, and obviously in 2019. So because we are living through this ongoing global pandemic that is the result of one of these RNA virus spillover events, I don't think I really need to emphasize the destructive potential of zoonotic viruses or convince you why studying their infection is important. But I would like to address what I see as maybe one of the more pressing questions surrounding zoonotic viruses, which is how do we begin to protect ourselves against these viruses that appear out of nowhere and then continue to shapeshift and trick our cells once they arrive? The first step, in my opinion, is understanding the strategies that they use to stay so adaptable. But this is challenging because, like I said before, one type or strain of virus can use many different strategies, and those strategies may be constantly changing. So really the first step is finding a better way to study populations that are so incredibly diverse. And this is really the goal of my research, which is kind of a interdisciplinary mix of virology and biological engineering. So I'm attempting to overcome the hurdle of studying these really heterogeneous populations by looking at viral infection at the single cell level. And What this means is that instead of looking at how a large group of cells responds to infection by a large group of viruses, I watch what happens when just one virus infects one cell. And the way I can do this is by physically isolating single infected cells into their own tiny bubble using a technology called droplet-based microfluidics. And the basic idea is that a microfluidic device is made of tiny micron-sized channels. And if we flow an oil solution and a water solution into these channels, at the point at which the oil and water meet, a tiny drop forms when the oil surrounds the water. The cool part is, because cells are made mostly of water, we can put almost any biological agent into this water solution, like infected cells, and create a drop that has just one cell in it. We can then use these drops to conduct mini experiments that are usually done with a large bulk population, but we can do them on just one cell at a time. So basically, by separating the members of the team, we can better understand what each of the strategies used by the individual players are. So how does this relate to viral infection? One of my projects is asking the question of how many viruses are produced during the infection of a single cell. So I'm using influenza viruses to answer this question. And it's important because, going back to our game analogy, if the opposing team has more players, they will likely be better able to, let's say, capture the flag that we are trying to protect. So to investigate this, basically what I do is infect cells with influenza, isolate those cells into a drop, allow the virus to replicate in just one cell at a time. And then after the virus has replicated, I can split off part of the drop that contains only the replicated virus, melt them down essentially to access their genomic RNA, and then mark that RNA with a fluorescent molecule. I then use a system of lasers to determine the fluorescent intensity of each drop, which means that if I shine a laser at a drop that has these fluorescent molecules, they get excited and make the drops glow. And whichever drops are glowing brightest contain the most tagged RNA, which tells me how many viruses were present in that drop, and thus how many viruses were produced during the single cell infection that drop came from. The most interesting part is then comparing thousands of drops that have virus produced from a single cell infection to determine if every cell in the population is replicating virus at the same rate, or if certain cells are pumping out more virus than others. And what we have found so far is that there is a lot of variability in how much virus is produced by seemingly identical cells. In our influenza experiments, we see virus production from a single cell that spans about 4 orders of magnitude, which is a lot. And that different strains of influenza viruses are replicating in the same cell type at different rates. And these results lead to some further questions about what characteristics of both the virus and its host cell create the optimal conditions for viral replication, and how we might use that information to determine the best defensive strategy that humans can use against these all-star opponents. And with that, I will take any questions that you have about viruses.
I actually had one question, Mallory. I was wondering, like, about your opinion about, so you were doing your study on single cells, right? But like I was wondering, what is your prediction of like, you know, like cell are in relation together, like they do mechanotransduction, they like send signals and cues together and stuff. So like I was wondering, like, because it's super interesting what you guys found about like single cells, I was like wondering, like, how do you predict if you like basically generalize your finding to a whole bunch of cell, like what's happening in our bodies? Like, how do you predict that they'll shine there?
Yeah, so when you're infecting like a large population of cells, you pretty much expect that whatever virus is reproduced in a certain cell is then going to go to the neighboring cell almost immediately and continue to reproduce. So in these large populations, there are more viruses being produced, usually on average. But something we're kind of interested in pivoting towards is like what are the genetic characteristics of those viruses. If they're going into neighboring cells, does like the end virus population have a different kind of genetic makeup than viruses that only have access to like one cell at a time? So yeah, good question.
I was wondering, you had mentioned there's a lot of specificity on strain type and cell type. What are the future implications of that if it's so specific to each individual RNA virus?
Sure. So I guess this is really specific to doing these experiments like in vitro in a lab. So there are like certain cell types that are pretty much model systems in labs that we use because we know that we can like consistently get successful infection of these cells. One of the cell lines we use a lot is actually like a canine cell line. So to like really translate that into more of an applicable system, you'd probably move to some kind of mouse model. But viruses in general are pretty specific to at least cells of a certain species. There will be some kind of like mouse adapted, you know, influenza virus or bird avian influenza virus that maybe wouldn't infect humans.
This is a little bit more of a sort of a broad, how does it feel question, Mallory, but like, how does it feel to have had a thing that you were working on and a sort of in a kind of a like, I'm going to graduate school to study science, and I have, I think I fixed on viruses, and to have it, you know, over the past 2 years become such a massive sort of cultural marker. I mean, they were always there and they were always important, but like this is essentially one of the primary things that many of us are talking about on the daily. How does that feel?
Yeah, it was an interesting timeline for me because so I'm in my second year, which means I joined my lab about this time last year for influenza projects. And then because we study viruses, as soon as there was like enough research about coronaviruses to start any projects, we pretty much pivoted like all of our projects for the next 6 months towards SARS, which ended up being a really cool learning opportunity for me. Now I've gotten to do some work in the BSL-3 lab on campus with the SARS virus. So as a, I guess, person studying virology, it's been a pretty cool year for research, but it has been a little hard to like separate what I'm thinking about for research with like what's going on and what everyone has just been constantly talking about. So the work-life balance is maybe not there.
Can you tell the uninitiated what a BSL-3 lab is?
Oh, sorry. There are different safety levels of laboratories that work with any kind of like biological agents like bacteria or viruses. And depending on how harmful that could be, if you were exposed to it, you have to take different precautions and like the protective equipment that you use and wear. So the SARS coronavirus that causes COVID is considered to be a safety level 3 agent, which means that you work with it in a facility that is has negative air pressure. You have to wear these like big Tyvek space suits essentially. Like all the air you breathe is HEPA filtered. Nothing can like come out of the facility without being like totally blasted with heat and chemicals. And yeah, so that's fun. Cool. Well, thank you.
Thank you, Mallory. Oh, here's another question coming in under the wire. How do you see your research impacting drug development? Will these drugs become more specific to each person's own body, or will you be able to generalize these drugs?
Yeah, that's an interesting question. Personalized medicine is definitely something that is like talked about a lot in my field. More in the sense of like screening your own genome for what would maybe be best suited to your body. So I don't think my research would like directly impact drug development, but we do have some people that in our lab that use the same drop system to look for antibiotic resistant bacteria, basically as a hopefully a way for doctors to better screen whatever infection you have and determine what antibiotic would be the most effective against that particular infection. Yeah.
Great, thanks, Mallory. So we have one more speaker today, and that is, sorry, I just tabbed out, Hazal Vahidi. Take it away.
Thank you, Kent. Hi, everyone. My name is Ghazal, and I'm a second-year PhD student in Mechanical Engineering department. I'm sure you've all heard about bone fractures without a trauma in older adults, like breaking a bone when just normally walking, or in some cases, even during a sneeze or a cough. Well, we call these bone fractures without a trauma bone fragility fractures. And in the United States alone, more than 2 million elderly people a year experience a bone fragility fracture, which basically costs more than $2 billion in healthcare. So as you see, this is a major health burden worldwide and one of the leading causes actually of mortality and disability in older ages. But what does happen to bones in aging? Like, what do bones become less strong and experience bone fragility fractures? Well, this is what we're going to explore together in the next 10 minutes. Well, resistance to fracture is determined by both bone quality and bone quantity. Bone quantity means how much bone is present, and bone quality talks about the quality of that bone. Is it too porous? Is it too soft? Like, how is it structured? So for your bones to be healthy and resistant to fracture, it is important to have a balanced bone quality and bone quantity. And in aging, unfortunately, we start to lose both of these factors. But why do bones lose their quantity and quality in aging? Like, what oversees these in our bodies? To answer these questions, we actually need to zoom in on the bones themselves. Well, although your bones look like white dead rocks in your body, they actually live very active lives. They're constantly being broken down and built back up to maintain their health. And we call this process of like building new bone and resolving the existing bone remodeling. Bone remodeling process in our body is done by our bone cells. Some of the bone cells behave like a construction crew. They constantly build new bones. Some of these bone cells, on the other hand, behave like a demolition crew. They crawl along, discharging a cocktail of acids and enzymes and dissolve away the bones. Some of the bone cells are the chiefs. They basically sense everything going on in our bodies, and based on what our body needs, they command other bone cells to build or destroy. And these chief bone cells are called osteocytes. Well, osteocytes are basically in charge of everything going on in our bones. They not only command other bone cells to build or destroy, when it's needed, they started to do the construction job or the demolition job themselves, which we call this osteocyte direct bone remodeling. Osteocytes live in these tiny caves all around the bones, which we call lacunae. And lacunae are less than 10 micron in size, which is 1/10 of a single sheet of paper. So like they're tiny, they're very, very tiny. And the other thing is osteocytes communicate with each other and with their construction crew and demolition crew through really complicated networks to make sure our bones are healthy. Imagine when the body is in urgent need of calcium, like when a mother is breastfeeding, osteocytes sense this and command other bone cells to dissolve bone and release calcium back into the body. And if it's needed, they even start their direct remodeling and dissolve their local bones. Another example, when you finally decide to go back to gym and start weightlifting, well, osteocytes sense this and tell other bone cells to start building new bone for you so they can compensate for your increased activities. Again, if it's needed, they work side by side with their construction crew. Now, because of the bone remodeling, actually our bones stay just strong enough for the activities we do. For healthy bones, bone remodeling should stay in its natural pace, a precise balance of how much bone is forming and how much bone is dissolving. And so you see the interplay between bone fragility and bone remodeling is actually complicated. And osteocytes need to make sure that the bone remodeling is in its balance, and therefore our bone quality and bone health and strength all are in good shape. So again, for a healthy bone, osteocyte need to make sure the bone remodeling is in its perfect balance, not more and not less. Now, when we say bone become more fragile and lose their strength in aging, is that because of an unbalanced bone remodeling, you think? Well, most certainly, yes. But do we know why exactly an unbalanced bone remodeling occurs with advanced age? That we don't know yet. But we suspect it is because of the osteocytes, whether these chief bone cells are dead or retired from their chief position, they don't do a good job of maintaining the bone health anymore. Now, a growing body of work, like scientists from all around the world have started to research osteocytes in aging. But mind, this is not an easy task. Remember, the lacunar size was about 1/10 of a single paper thickness. So we need ultra high resolution instruments for any study involving osteocytes. And we do have state-of-the-art instrument at MSU available to us, so it will enable us to study osteocytes. The other thing that makes this work a little bit challenging is the factors that complicate the bone remodeling. Let me ask you, do you think bone remodeling and its complicated relationship to bone health is similar among males and females? What about in people with different genetic background, or people with chronic conditions or different diseases? Well, all valid questions and all most certainly affect osteocytes' function. But we must start somewhere, and a good strategy is to start simple, understand the basics of what's happening to osteocytes in a normal aging, and then continue from there. So in my research, I use a mouse model of aging. Well, mice are the smallest organism that share physiological similarities to humans, especially in their bones. So there are popular choices for the investigation involving the bones and skeleton. We have male and female mice in 2 stages of the life. Young adult mice, like 30 years old human, and old mice, like 80 years old human. And an important part of my research is to investigate if these chief osteocytes are getting retired as we age, or they die as we age, or both. So my research has 2 main pillars. The first pillar is to assess if there is less osteocytes with aging, meaning if they actually start to die. For this, when the mice are alive, we inject them with special safe dyes, and these dyes are designed to color the osteocytes so they can shine in their habitat of lacunar voids. Makes it really easy to visualize them later. So we euthanize the mice, extract their bones, employ different combinations of ultra high resolution instruments to see how many osteocytes are still living in their lacunae. So basically, an empty lacunae is telling us that my resident osteocyte is dead. Now, interestingly, we actually see less osteocytes with aging. Yeah, these chief bone cells start to die in advanced age, and this is one of the reasons for an unbalanced bone remodeling. The next pillar of my research is to assess if the osteocyte activity is declining with age, basically if this chief starts to retire. For this, we again inject mice with special fluorescence labels that are designed to show us the path of bone remodeling. This is basically like giving stain tools to construction or demolition crew or the osteocyte themselves. So basically, if they use those stain tools to build new bone or destroy bone, we will be able to later track them based on the marks they're leaving behind. And we use different imaging techniques to capture these changes, these marks. And then we generate codes and use computer-assisted processing to study the amount and rate of the bone remodeling. Here again, we suspect to see less bone activity. Basically, we think these chief bone cells are kind of losing their cell reception. So they start communicating less with their construction and demolition crew and also do less hands-on job, which leaves the bone remodeling super wacky. Now, to get a complete picture of what happened to bones in aging, there are many other stuff that we do on these bones. We test their strengths, we test their composition, their structure, and many more. But at the end of the day, this is what we think that is happening. We age, some of these chief osteocytes die, and some retired. Basically, they lose track of what is happening in our bones, and therefore other bone cells lose their motivation and guidance for their construction job and demolition job. As a result, both bone quality and bone quantity decline, and bones start to weaken and fracture during normal activities. Now imagine if we could solve the mystery of osteocytes, if we could understand how we could prevent the osteocyte death and inactivity with aging, then that would enable us to design osteocyte targeted therapeutics or even predictions for the bone fragility in elderly people, so the bones can remain healthy and strong in the golden years of the adulthood.
Giselle, I have a question.
Sure.
So you mentioned that you use these mice that are kind of like 80-year-old people. How old are the old mice?
They're actually 22 months, which is a big deal in mice lives. So they're actually pretty old, and it kind of makes it hard to work with them because we lose a lot of them during the experience because of the natural causes, and then their bones are super fragile, so we have to be extra careful.
Yeah, so do you actually create cell lines of mice too? Do you have to wait like 22 months for these specialized mice to age?
It's actually a very good question. So basically, when we get NIH grants for our labs, they kind of provide us with this age mice. So we tell them what kind of strain of mice we are willing to use, and they basically send us this pretty old mice. And yeah, we kind of can tell them what age range is works the best for us. Like if we want to give them treatments or special diets and everything, then we can design based on that. But we get them from NIH laboratories.
So cool. Thank you.
Sure.
I don't know if you've been able to get to this point in research, but I was wondering if it was possible to look at if the lesser down bone builders could take the place of a chief builder. if one of them dies or not, if that was possible?
That's actually a really good cool and sophisticated question, because I didn't go into the details of like where these chief bone cells come from. Interestingly, there is a transition from these builder cells to chief cells that happens in our body during our growth. So we have these two different cells that are kind of like born with us, that are like builder cells and destroyer cells. And then as we grow, like during those like very first days in our like basically we're infant, like we're during those very first days that our bones and skeleton are shaping, these some of these builder cells become these chief cells that they promote kind of. But it's not like, unfortunately, like as we age, our cells also lose that ability to become as osteocytes, like for the builder cells to become osteocytes. But osteocytes, believe it or not, are kind of their potential and what they can do in our body are newly discovered. Like they've been around for more than 50 years, I don't know, but it's not more than like 15 years that we understand these like chief bone cells. Not only they command and manage other bone cells, but they also do both of the building and destroying. So there's a lot unknown about these cells yet. And yeah, we were excited to discover what's going on with them.
I just had a quick question. You said you don't know the exact reasons why the osteocytes lose their commanding ability with age, but what are some of your theories as to why that happens?
Yeah, another good question. So it's actually like, to answer this question, I should first mention I'm just a mechanical engineer exploring the biology world. So I'm really new to this kind of stuff of like what exactly happening like behind the scene. But what we think is first, so osteocytes are dying, which makes sense. Like if they die, there is like less of them and they cannot do the job anymore. But for their decreased activity, like basically what keep osteocytes active, it's about our blood cells and like what's happening in our neurons, because these osteocyte cells should get signals from other parts of our body. Like for example, if you're falling and you're breaking a bone, like first, like these bone cells should get signal from your brain and from other parts of your body, from your increased blood flow to them, increased fluid flow to them. And so they're getting signals from all around the body. So one accept would be like when like as like the rate of everything is slowly decreasing in the body, they're gonna get probably less signal from their outside environment. But also they have really sophisticated mechanotransduction system on them, like their cell receptors are super sophisticated. And we think as we age, like these cell receptors are kind of losing their sensitivity, so that causes the decrease the activity. So these are some theories that are out there, but it's a work in progress, honestly.